Hello, welcome to the Upadhyay Lab for Cellular Biophysics, where we study how physical forces enable a cell to sense and respond to its physical environment. We use physics concepts and measurements to obtain insight into the fundamental mechanisms that govern biological systems. Our research focuses on two main areas: mechanical regulation of the immune response, which helps understand how our body fights off infections and cancer, and mechanical regulation of gene expression, how genes are turned on and off. To study the physics of these living systems, we use high-resolution optical microscopy and computational image analysis to examine the inner workings of cells. We image and quantify the nanometer scale dynamics of proteins and cells. We measure nanonewton forces generated by cells. We examine how these are perturbed in diseases, and we build physical models to understand underlying mechanisms. And now for a virtual tour of a typical day in the lab.
In our lab, we study the mechanical regulation of immune cells. Immune cells have a skeleton called the cytoskeleton, which is made up of biopolymers, actin, and microtubules, as shown in these images. These biopolymers allow immune cells to generate cellular forces and allow immune cells, such as the one in orange, to contact cancer cells, such as the one in blue, exert forces on them, and kill them.
In collaboration with Dr. Gordon Hager's lab at the National Cancer Institute, we work on understanding how the physical environment of a cell influences gene expression. At the single molecule level, we track individual transcription factor molecules as they search for their target sites in the genome. On the length scale of a single nucleus, we look at the mobility of chromatin using polymer physics models. And at the multicellular aggregate level, we measure traction forces exerted by cells as they interact with their surrounding.
We measure force generation by immune cells by imaging cells on gels embedded with fluorescent beads. As cells exert forces, the beads are displaced from their original locations. We then use the theory of elasticity to calculate cell traction forces from these displacements.
From our single particle tracking data, we use statistical physics models to quantify transcription factor dynamics across time and space.
I would like to thank all the amazing people in the lab that have made this work possible and create a vibrant and intellectually stimulating atmosphere. Our research is supported by funding from federal agencies and benefits greatly from collaborations. Please contact us if you're interested in doing cutting-edge research at the interface of physics and biology. Thank you.